Welcome, everybody. Thank you very much for joining us today in one of the lunchtime seminars, perhaps one of the more unexpected topics as you've gone through the symposium and conference here. Today's session will focus on the expanding roles of oligodendrocytes, both in normal brain biology as well as in disease states. And so I say unexpected because when we think at a conference like this of CNS gene therapy, we primarily think of targeting neurons. And of course, we all recognize that the brain is composed of many other different types of cells, all of which have critical functions, but among them notably are these oligodendrocytes. And yet we haven't really focused on them as a field in terms of understanding their multiple functions in the brain, looking at them from a drug development standpoint in terms of how these cells can be targeted, and understanding more deeply the roles that they may play not only in the usual suspect kinds of disorders like white matter diseases, but in broader brain diseases. And so we wanted to take the opportunity today to have a session and bring together a group of experts who have focused on these topics during the course of their careers. People who have studied normal biology of oligodendrocytes, understood their multiple functions, and investigated their roles in disease states. I will introduce each speaker as we go through the session one by one. And just as a point of order, we will wait until all the speakers have completed their presentations and then invite them back up and do Q&A for all the talks at once at the end of the talks. So maybe just to set the stage a little bit, I think many people here are probably generally familiar with these really fascinating cells, oligodendrocytes. And we're familiar with them from the standpoint of what they have historically been understood to do, which is make myelin. And myelin is the key insulating material in the brain that helps neurons function. And so we've always looked at them as a cell that is really supportive of overall brain function, and particularly neuronal function. They kind of help facilitate the action potential and propagate the signal all the way down the axon of the neuron to the synapse. And that was obviously a very important support function that they carry out. But, you know, nature's pretty crafty and tries to maximize the output and capabilities of different cells. And over the course of time, it became known that these cells also carry out important, let's say, metabolic functions, producing growth factors, trophic factors, providing metabolic support, including being a major source of the components that go into ATP production. And so critical, we all know how energy dependent, how energy intensive the CNS is and neuronal function is. And so having an adequate supply of ATP for energy purposes is also central. But then things get really interesting because we start to see that these cells may play more broader functional roles, that they may actually participate in the development of synapses. They can help shape and carry out synaptic plasticity. They can prune neurites. They can increase the intensity with which synapses function or decrease them. And so all of this unifies into a deeper understanding, which is now coming through the literature, where these cells may be actually instrumental in memory and learning and in broader cognitive functions than was originally anticipated or thought. And so for this reason, we think that they really represent a great target to be looking at, certainly under-investigated when you think about the comparative effort that's gone into neuronal investigations for therapeutic purposes. And that leads us then to, well, where could these cells potentially play a role? So obviously, the usual case to think about would be leukodystrophies, so-called white matter diseases. The other place would be disorders of myelin, like MS, where there's a disruption either in myelin production or a loss of myelination and demyelination of the axons. But strikingly, even more recently, we've seen that it kind of expands beyond that. There's this ability for these cells possibly to play a role in broader diseases like Alzheimer's disease, Huntington's disease, and epilepsy. Now, these things have to be proven out, but they represent really interesting starting points when you think about why have we had so much trouble developing effective therapeutics for intractable CNS disorders. You know, as scientists, as physicians, we think about, well, what should we be looking at? And we've kind of self-selected that the neuron is the one cell that we really should target. But we really should probably be thinking more broadly than that, because we have not made tremendous progress, at least the expected progress, in developing therapeutics for this broad range of other disorders. And so coming out of this meeting, we hope you learn a lot about oligodendrocytes, and maybe the provocative question is, should these cells, and others as well, have they been overlooked as potential targets in drug development and therapeutics development for these intractable CNS disorders above and beyond myelin-based disorders and white matter diseases? And so it's my pleasure now to welcome our speakers. We'll begin with Ethan Hughes. Ethan is Associate Professor in the Department of Cell and Developmental Biology at the University of Colorado School of Medicine. So Ethan, please come up to the podium.
Okay, thank you very much for the kind introduction, as well as the very nice introduction to the importance of oligodendrocytes in myelination, not only in health, but also in disease. And so in this short talk, I'm going to focus around the role of oligodendrogenesis and myelination in the context of the healthy brain, specifically around motor learning. Now, my talk's going to be structured into 3 different parts. First, I'm going to continue the introduction of oligodendrocyte lineage cells. Then we're going to move into a section focused around oligodendrogenesis, or the production of new myelinating oligodendrocytes in the CNS in the context of motor learning. And then lastly, we're going to focus on specifically the effects on myelination of motor learning in the healthy brain. And the big goal here is really to understand the fundamental mechanisms that really regulate oligodendrogenesis and myelination in a healthy brain, to be able to potentially translate these insights into the context of neurological diseases, that we may be able to apply some of these insights moving forward clinically. Now, as you just heard, oligodendrocytes are the myelinating cell of the central nervous system. They extend multiple processes out into the tissue, where they wrap around neuronal axons, forming this multi-laminate structure of a compact myelin sheath. And so the function of myelination in oligodendrocytes in the healthy CNS is to accelerate potential propagation, reduce the energetic costs of synaptic transmission, as well as, you heard, provide metabolic support to neuronal axons. And oligodendrocytes are generated through a process of differentiation from an oligodendrocyte precursor cell. These precursor cells are quite interesting. They tile the entire central nervous system, and they continue to produce new oligodendrocytes throughout life, not just in the early developmental phases when myelination is most known to be dramatically increasing. Now, the focus of the talk today is really around this new concept of myelin plasticity, which may be an important role that the brain uses to modify brain circuits, as well as learn new tasks. And there are several different aspects of oligodendrocyte lineage cells that can be modified by neuronal activity. First, the differentiation of these precursor cells. Also, neuronal activity and changes to neurons can change myelin thickness, or the number of myelination wraps. They can also modulate the length of the myelin sheath itself, or even the number of myelin sheaths that an individual oligodendrocyte can make. And then it also can dictate which axons actually receive this myelination themselves in terms of an axon selection. And just to introduce a main technique that my lab uses to study these concepts, in that we combine head-fixed in vivo imaging using multiphoton microscopy with genetic models that label different cell types with EGFP or other fluorescent proteins. And using this cranial window-based approach, we have unlimited access to visualize individual cells and subcellular structures from minutes to months, and we can actually watch their dynamics to different perturbations to the animal. And so now I'm going to move into this motor learning section of how does learning a new motor task actually modulate the formation of new oligodendrocytes. And as I mentioned, this process is a dynamic process, starting with oligodendrocyte precursors and moving through the stereotype differentiation process to form new myelinating oligodendrocytes. And the field has some pretty amazing work in this space that set up these questions. First, work from Michel Mongi's group has shown that channel rhodopsin expression in neurons and subsequent driving neuronal activity actually drives the differentiation of oligodendrocyte precursors to form new myelinating oligodendrocytes. And further work from Bill Richardson's group has shown that if you block the formation of these adult-born oligodendrocytes, this actually impairs the ability of mice to learn a novel motor skill learning task. This work has been followed up by 2 other groups recently, showing that this is not just important for motor learning, but also memory consolidation and the formation of long-term memories. And so we really wanted to look at how the dynamics of this process was really regulated within an animal situation. And so to do this, we trained our mice on a forelimb reach task where they reach through a slit in the plexiglass box for a food reward. They can get quite good at this technique. This is an example of one of these mice performing this task. And this movie is slowed down by 10 times. And so you can see that their behavior is very quick, very precise, and very cute. And so these guys take about a week to learn this task. And so over this period of time, we then can watch how oligodendrocyte lineage cells as well as myelination can change on the neuronal circuits in primary motor cortex. And so just to set you up with how our data is really interpreted, and so we can image mice every other day for extended periods of time, and then we can perform different behavioral interventions where we either have no activity, we can train them on the forelimb reach task for 1 week, or we can train them on the forelimb reach task and then watch what happens when they rehearse the task many weeks later. And in using these transgenic mice, the mouse that we're using specifically turns on EGFP just in the myelinating oligodendrocytes, and this really allows us to watch the formation of new oligodendrocytes here highlighted in green over the period of 30 to 40 days in vivo. And so what this allows us to do is that we can actually count the number of new cells that are generated in the same cortical volume, and we can calculate a cumulative cell gain across this period of time, and then look at how behavioral interventions like learning alter the rate of oligodendrogenesis in these different phases of the experiment. And when we did this, we found that, to our surprise, actually the formation of new oligodendrocytes was actually suppressed during the motor learning task, and then subsequently there was a dramatic increase in the rate of new oligodendrogenesis. This, to test whether this is just an effect of the novel task itself or just practicing the task, we looked at what happens when the mice just practice the task. And what we actually found is that there is a higher rate of oligodendrogenesis in the baseline period before they even begin practice, that then again tapers off over time. And so putting these 2 groups together, we found that oligodendrogenesis is actually suppressed during the acquisition of the learning process. And then there's a prolonged increased rate of new formation over the period of about a month following the acquisition of a new task. And so this sets up a lot of really interesting fundamental findings to understand of what is the function and of these newly generated oligodendrocytes and where are they placing their myelin and why are they required for this process to begin with. So to get into some of those questions, we're going to focus specifically in on myelination now and the response following motor learning. And so not only can we track individual oligodendrocytes in vivo in our mice, but we can also track the formation and, oops, let's try this one more time, the formation and pattern of myelin in this process as well. Could you double check this? Movies doesn't seem to be playing. Should be on the click. There we go. Is it going? No? You don't have the movie? Okay. Well, what you would see in this movie, which we'll have in stills later, is that each one of these fluorescent lines here is actually a myelin sheath. And we can both trace the myelin sheath as well as about 70% of these connections back to individual oligodendrocytes. And so we can define on a subcellular level individual myelin sheaths and how they change over time from one individual oligodendrocyte. And one other piece of dogma that we should all kind of correct is that it's historically been thought that myelination persists from the initial axon segment all the way to the terminal field of the synapse along the neuronal axon. But recent work from Paula Arlada's group has shown that in the cortex, at least, many axons can be intermittently myelinated, where there can be large gaps in between these individual myelin sheaths. And the function of this arrangement is currently unknown, but one possibility is that these axons could be templates for new sheath generation that may modulate the conduction velocity or spike timing of these neuronal circuits. So what we wanted to look at specifically is, does learning modulate the pattern of myelination along these intermittently myelinated axons in the cortex? And so again, we used our 4LM reach, and now we can look at individual nodes of Ranvier. These are the gaps in between myelinated sheaths. And what we found to our surprise is that myelin sheaths can actually retract, elongating these nodes of Ranvier, which are the sites of action potential initiation, and this is associated with learning. These are our in vivo imaging of the intact animal. Post hoc immunostaining also confirms that these myelin sheaths are along the same axon, and that these widening nodes or lengthening nodes only occur after the acquisition of the learning process, really kind of driving this potential new way that myelination can be dynamically modified by behavior. And we think that this is very correlated with the length of the myelin sheath. So we think that the myelin sheaths themselves are actually retracting, driving these lengthening nodes of Ranvier in these conditions. We wanted to look whether or not this was specific to individual oligodendrocytes. And so we looked at individual nodes of Ranvier or their myelination pattern along single axons in cortex. And what we found is that all oligodendrocytes' myelin sheaths have the probability or the potential to be able to retract and or lengthen. But these retracting and lengthening nodes seem to be occurring on specific types of axons within the primary motor cortex, really suggesting that this may be an axon-based phenomenon, that myelin is being modified specifically on these axons. To look at the functional consequences of this modulating myelination pattern, we collaborated with Ilan Polig-Polsky at the University of Colorado to model these effects. And so under normal circumstances, conduction velocity occurs quite quickly, around 2.5 meters per second. However, if we modify the pattern of myelination along these areas, we could actually find that there's increased amounts of delays, as well as some proportion of failures that can be generated with the lengthening of these individual nodes of Ranvier, really suggesting that these myelin dynamics may have functional impacts on the timing and conduction velocity of individual action potentials. Now, overall, we not only saw these cellular changes, but these same cellular changes of myelination pattern were correlated to the behavioral performance of the mice on this reach task, in that mice that were more proficient at performing this task actually had larger amounts of node lengthening, specifically during the learning phase, whereas we found, similar to our previous findings, that oligodendrogenesis, or the generation of new oligodendrocytes, mice that performed better on the task actually had more oligodendrocytes born in the post-learning period. So to summarize what I've told you today is that motor learning itself in the healthy brain can modulate the dynamics of the formation of new oligodendrocytes. Furthermore, motor learning can modulate the pattern of myelination on specific neuronal axons, as well as through both node lengthening as well as oligodendrogenesis. And in data I didn't have time to tell you today is that these axons are actually involved in the reach task itself. So it seems that this is an axon-specific learning-induced plasticity along individual axons in the motor cortex. And these changes in myelination pattern may really have important implications not only in conduction velocity and spike timing, But it also opens up a lot of other questions in that we know nodes of Ranvier have targeted voltage-gated ion channels, and so this may further affect axon conduction velocity. But it also opens up questions of what happens at axon branching or synapse formation in these newly exposed areas of axon. Could this open up possibilities of axon-axon signaling, as well as change the overall metabolic support that myelination is providing to these axons? Lots of questions to explore. And then lastly, we find that these cellular changes are really correlated to behavioral proficiency in these tasks, really highlighting that this plasticity of myelination on neural circuits is really important for learning new tasks as well as performing well on them. And so with that, I'd just like to thank all the members of my lab, specifically Clara Backmeister, who really drove a lot of this work. And at the end of the session, I'd be happy to take more questions about this project. Thank you very much.
Thank you very much, Ethan. Would now like to invite Dr. Woodrow Peelerts up, Assistant Professor at KU Leuven, who will talk about targeting oligodendrocytes in neurodegenerative diseases. Thank you, Woodrow.
Thank you. All right, switches here. All right, so for this part of the session, I'll be discussing some of the latest developments and some different modalities for targeting oligodendrocytes in vivo, so both for experimental modeling as well as for new therapeutic strategies for targeting oligos in vivo. So first, I'll just start with a quick introduction to oligodendrogliopathies, primary oligodendrogliopathies, some of the different modalities that have been used over the last couple of years. We'll be discussing also the types of capsids and promoters that have been used to drive specific expression in vivo and specific targeting of oligos in vivo. And then also finally, I say a few words about modifiers of oligodendroglial tropism, so discussing a bit about injection timing and as well as administration routes and how this can affect oligodendroglial tropism. So focus here obviously are oligodendrogliopathies, and so this is, I believe, an expanding spectrum of diseases in which you'll have the leukodystrophies, encephalopathies, but also, for instance, synucleinopathies or tauopathies. So it's an incredibly wide range and an incredibly wide spectrum of diseases that are incredibly heterogeneous, which can arise because of genetic causes, but they can also be acquired. They can be common or rare, with incidences between 1 in 5,000 to 1 in 100,000 people. Most, if not all cases of people with primary oligodendrogliopathies are treated symptomatically. Mostly oligos are affected, but especially, for instance, in leukodystrophies, astrocytes or macroglial cells, or even different neuropathies, or even a blood vessel pathology can lead to myelin damage and oligodendroglial dysfunction. So these oligodendrogliopathies can happen at any stage during life, but usually the earlier they arise, the worse the disease outcome will be. And so in our lab, we're interested in the disease mechanisms and how these different diseases arise. So we're using viral vectors here as a tool to develop new animal models and to really understand how these diseases come about. So developing new preclinical animal models for different types of oligogliopathies. And then, of course, also we're interested in developing new modalities or developing new strategies to really target oligos in vivo, as, of course, there's also a need for better targeted therapies. And so some of the strategies that have been used over the years is by either through rational design of new viral vectors, so combining different capsids and promoters, and either restricting, for instance, expressions to oligos in vivo. Capsid shuffling, for instance, has also been used to target oligos and as well as peptide display. And so many of these viral vectors have been delivered intravenously, but most of the applications are still focusing on direct injection, intracerebral delivery, either through the lateral ventricles, cisterna magna, or just by injecting them, for instance, in the striatum or myelin-rich regions. And so here, I'm just showing an overview of all of the different serotypes that have been used. And you can see there's quite a big range and variety of viral vectors that have been used to actually target oligodendrocytes, sometimes in combination with constitutive promoters, but also very often in combination with cell type specific promoters such as myelin, MEP, MAG, CNP, and other hybrid and synthetic promoters as well that have been used to selectively target or to restrict expression of different transgenes of interest to these cells. So one of the first studies that I actually looked at targeting oligos in vivo was a study where they used a broad tropism AAV serotypes, AAV capsids, but then in combination with cell-specific promoters, here in this case the MVP promoter, to really restrict that expression to different cell types. And so there in the top panel here, just use this here. So in the top panel here, you can see a GFP expression, transgene expression after injection here into the striatum. You can see expression here restricted to neurons by using the CAG, a constitutive promoter, but then by just switching the promoter to an MVP promoter, you can see expression here in the striosomes, in the striatum there, for your different types of oligodendrocytes within that area. And so by just simply switching here different promoters, so for instance the CAG, CMV promoter, you can see that you get mostly expression here in your neurons with a very high specificity, but then by just simply substituting the promoter here, GFAP or MVP, you can see that you can target or at least restrict transduction or expression in either astrocytes or oligodendrocytes. Another example here is another broad tropism AAV capsid, the AV21, same strategy here by using again the MVP promoter and restricting expression to oligodendrocytes, where again you can see that you get relatively high specificity for these cells in vivo. Another interesting strategy was by developing new type of promoters, and so this is from the Pleiades project where the authors here then looked at regulatory regions, for instance, for OLIG1 or OLIG2 cells, and basically cloned and synthesized their own synthetic promoters and use those promoters then again to drive cell-specific expression. And so one of those promoters that was identified was the PLE151 promoter that after knock-in here actually drove specific expression in oligodendrocytes in the cortex and in other regions of the mouse brain here. And then afterwards, in a follow-up study, these sequences were then again cut and shortened so that it can also be used for as a promoter in viral vectors, and that you can see here after intravenous injection again, you can see again that different cell type, or different modalities, different constructs can actually drive specific expression in oligos in vivo. And so we've used a similar strategy by combining broad tropism AAV capsids and then using cell-specific promoters such as the MAC promoter or the MVP promoter. And again, by doing so, you can see that after injection into the striatum, in this case in rats, you can see again that you get very high specificity of transgene and transduction here again in oligodendrocytes in vivo. You get still some expression in neurons. We also played around with some hybrid promoters, so combining, for instance, an MVP promoter with CMV intron, for instance, to get more robust expression, but that mostly leads to a reduction in specificity. So that I think summarizes the work that's been done by combining again these broad tropism capsids with cell-specific promoters. And so another question is obviously whether you can more specifically target oligos by just changing the capsids and then using different types of capsids. And so this is also some work that we've done here by just substituting the serotypes and then looking at how different serotypes can influence targeting and transduction of your oligodendrocytes in vivo. So first what we did here is to again look at expression specificity. And just by simply using the 2.7, 2.8, or 2.9, we didn't notice any obvious differences, so the specificity is actually maintained. But what was really obvious is that some serotypes have very different expression efficiencies and transduction volumes. For instance, the 2.9 leads to a tripling of targeted cells and transduced cells in vivo, which also translates to a larger transduction volume. And I think you can also appreciate here from those serial sections here that, for instance, the 2.9 gets really broad expression and widespread expression in the striatum, where, for instance, for the 2.8, this is actually much lower. And then following this, I just want to mention, or just want to say a few words about the PHBB capsids developed by the Gradinaru lab, which I think is also a very interesting system, or a very interesting serotype to target oligodendrocytes. So these were generated through inserting a heptamer amino acid residue, I think in the VP1 region of a 2,9 serotype. And so they developed a large library and injected these intravenously in Cre transgenic mice, only expressing Cre and astrocytes. So through targeted or to directed evolution, then this library was screened for their ability to first cross the blood-brain barrier, but also to transduce glial cells. And so what you can see here is from one of the, I think, really interesting candidates that came out of this screen is the AVPHBB serotype. You can see that this very efficiently transduces astrocytes as well as neurons, and so in this context here, also oligodendroglial cells. And so this was then, this capsid was then further improved, so there's an enhanced version of this one here. And so then colleagues then at Bordeaux, Erwan Bezard and Benjamin Dehaye, then also used this type of serotype to look at transgene expression in macaque, non-human primates. And so they injected intrathecally to examine again the tropism of this specific serotype, the specific capsid. And they also noticed again the broad tropism of this capsid, again transducing oligodendrocytes, neurons, astrocytes, as well as microglial cells. Kind of interesting there as well. And so then again, by using those cell-specific promoters, for instance, the MBP here or the MAC promoter here, you can again restrict the expression to oligodendrocytes. And here in this example, you can see how different viral vectors were then mixed and combined and injected into the same animal. And by using different cell-specific promoters, you can see that you can get specific expression in astrocytes, here in neurons, oligodendrocytes, and then just combined, you can see those differentially expressed reporter genes. We've also used this in human oligodendroglial brain organoids to see if we could also transduce and target human oligodendroglial cells in those organoids. And so again, I think you can appreciate it by using the MAC promoter here that we can again restrict expression to those oligodendrocytes. And so in human oligodendroglial myelinating cells, we also know that this specific system works efficiently and that we can target these cells in this particular setup. And then a last example when it comes to a capsid is the AAV-OLIG1, and I'm sure you'll hear much more about this. So this serotype was developed through capsid shuffling and injecting again this library of viruses intravenously, I think in rats. These rats were then also treated with 6-hydroxydopamine, meaning that there will be degeneration of the dopaminergic neurons and degeneration of the striatal axons in the striatum. This also results in some proliferation, oligodendroglial proliferation in the striatum. And so by, again, injecting this library, the authors were able to select a serotype that targeted specifically oligodendrocytes in the striatum. And so by isolating again from the striatum, they found the OLIG-001 serotype that binds specifically with oligodendrocytes, and where you can just use a constitutive promoter here, the CBH promoter, again to drive robust expression in vivo. And this is just another example from the lab at Jeffrey Cordover, where they also used these viral vectors to target the caudate nucleus, the putamen in non-human primates. And so by directly injecting and targeting these areas, you can get, again, robust expression and widespread expression here in the striatum. They have also used this, for instance, to overexpress alpha-synuclein, which is a central protein in multiple system atrophy, Parkinson's disease, and other synucleinopathies. And again, by just using this system here and overexpressing the protein, they were able to actually develop a non-human primate model of multiple system atrophy, which I think was really impressive. So then just finally, a few words about some modifiers and administration route and injection timing, because those apparently are also or also have an influence on the tropism and the distribution of these vectors. And this hasn't been studied in a systematic manner, but there have been a few studies looking at how, for instance, administration routes and timing can influence the tropism of these vectors. Here, for instance, you have the AAV1 viral vector, with the MVP promoter that was injected in either P0 or P10 and P90 animals. And you can see that at the P0 stage, that when injecting these vectors into the striatum, you get mostly transduction of astrocytes or expression in astrocytes using this system. But then later on, when you go to P10 or more adult stages and again inject them in the striatum, you can see that you get this cell type specific expression again, and that you're targeting oligodendrocytes. So we've done a similar study where we also looked again at injection time and different administration routes. So for instance, here in this case, we've injected in the lateral ventricles of neonates at P0, P1, and P2. And again, here you can see actually big differences between different brain regions. For instance, here at P0, you can see some astrocytes positive here in the cortex. In the olfactory bulb, you see a lot of neurons, but also here in, for instance, the hippocampus and other areas in the brain. But then when moving to P1 or P2 neonatal stages, you can see that this pattern completely changes. Hardly any neurons that are detected. And in the hippocampus and other regions, such as the pons, the midbrain, spinal cord, we mostly see oligodendroglial specificity. All right, and just to wrap that up, I think what this shows is there's an extensive toolbox available depending on your scientific question or your therapeutic strategy that I think can be used to target oligos in vivo. There are multiple serotypes that can give functional transduction in oligos, and you can, for instance, try to restrict expression through using cell type specific promoters. Or, for instance, using cell-specific or targeting capsids, such as the OLIG-01, that can specifically target oligodendrocytes to get more and robust expression. Something I didn't really touch upon, but which has also been shown here, is that there are capsid-promoter interactions, especially for oligodendrocytes. So I think every system really needs to be tested to make sure that there is functional transduction and specificity for those different vectors. And then timing and administration routes can also influence tropism and expression specificity. So also they're important things to bear in mind. And with that, I would just like to acknowledge everybody involved here, and I would like also to thank everybody in the audience for your attention. Thank you.
Thank you very much, Wouter. Appreciate the talk. Next, we'll invite Chris Jansen up to give his presentation. Chris is a neurologist at Dayton Children's Hospital and Ohio Health Neuroscience Center, and he is also the co-PI on the Canavan disease phase 1/2 gene therapy first in human clinical study.
Thank you, Mark. So good afternoon. I'm Dr. Christopher Jansen. I'm the PI on gene therapy for Canavan disease, CAN-GT, which is sponsored by Myrtelle. And I'm pleased to present the interim 6-month data from our ongoing study. So first of all, I'd like to briefly discuss what is Canavan disease. So Canavan disease is a rare genetic neurological disease with devastating clinical consequences. It's categorized as a pediatric leukodystrophy, meaning that the brain white matter does not properly form and in fact is destroyed over time. Clinically, Canavan disease is distinguished by 3 cardinal features: macrocephaly, or an overly large head, poor head and neck control, and severe psychomotor retardation with inability to walk, talk, or basically perform any age-appropriate behaviors. Currently, there is no FDA approved drug treatment. So Canavan disease was named after Myrtelle Canavan, a Harvard neuropathologist, and it seems appropriate that the gene therapy company Myrtelle was named in her honor. Now, what she described approximately a century ago was a spongiform pathology of the brain where the brain became waterlogged like a sponge. Van Bogert and Bertrand were Belgian neurologists who subsequently showed the pathology involved myelin and white matter exclusively. But it wasn't until the 1980s that Kvittingen first described the role of elevated NAA in Canavan disease, what turned out to be Canavan disease, and the genetic defect was discovered by Madelon and collaborators in the 1990s, thereby enabling gene therapy to happen. What we now know is that Canavan disease is caused by mutations in the ASPA gene for aspartoacylase, an aminoacylase enzyme which breaks down NAA into acetate and aspartate. When this gene is missing or damaged, NAA increases to high levels throughout the brain. Effects include insufficient N-acetyl group substrate for myelin synthesis, as well as direct osmotic damage to myelin from local effects of high NAA. So just a little bit of history. I've been personally involved in drug development for Canavan disease for over 20 years now. In 2001, with my colleague, Dr. Paolo Leone, we conducted the first clinical gene therapy for Canavan disease with an AAV2 ASPA vector. And this was the first application of AAV for a neurological condition. At the time, there was no vector specifically targeting oligodendrocytes. The development of an AAV vector with tropism for oligodendrocytes took about 16 years through capsid mutagenesis studies by Steve Gray at UNC, who also joins us today. AAV-olig was really a breakthrough as the first AAV vector showing strong tropism for oligodendrocytes through capsid-host interactions. As you know, most existing AAV, and as we just heard, most existing AAV vectors are strongly neurotropic. AAV-olig is unique in its ability, or nearly unique, to specifically target oligodendrocytes, which are the main cell type involved in Canavan disease. So the drug product has already garnered a number of key designations, including FDA Fast Track and Orphan Drug Designation and EMA Orphan Drug Designation. So there are a number of features of Canavan disease. Right here, which make it an ideal gene therapy candidate. Number one, it's a single gene disorder, so gene replacement therapy is feasible. Number two, there's a well-defined cell population in the CNS which can be neurosurgically targeted. And number three, as a consequence of the brain-specific targeting, a relatively low dose of vector can be used, which makes it less likely to result in immunological complications and scalable, so more patients can be treated. The AAV-olig vector shown here at the bottom expresses ASPA predominantly in white matter. And in mature humans, unlike in rodents or other species, this constitutes almost half the adult brain volume. So specifically, AAV-olig expresses ASPA in proximity to myelin sheaths, which should decrease local NAA and prevent osmotic damage while simultaneously promoting myelination. So for this first-in-human study with AAV-olig, we enrolled 8 patients who were treated with a fixed dose of 3.7 times 10 to the 13th vector genomes. We performed direct neurosurgical delivery to the cerebral ventricles and surrounding white matter using either 2 or 4 delivery catheters introduced through burr holes. In addition to safety, our primary clinical outcome measures are the gross motor function measure, or GMFM, and Mullen scales of early learning, both of which we selected in our original gene therapy study 20 years ago. In my opinion, these outcome measures are among the best available tools for detecting meaningful clinical outcomes in Canavan disease, and we have decades of natural history data to refer to. All of our surgeries and follow-ups are currently being done at Dayton Children's Hospital in Dayton, Ohio. Some of the secondary outcome measures include non-invasive imaging with synthetic MRI or psi-MRI, a quantitative and rapid method to measure white matter, myelin, and brain water content. In addition, NAA levels are precisely measured in the brain using MR spectroscopy. We're using quantitative modeling of NAA concentrations using LC model with standardization to a phantom. So study inclusion criteria include a definitive diagnosis of typical Canavan disease by a board-certified neurologist, sequencing for identification of gene mutations, and age between 3 and 60 months at the time of treatment. Some of the exclusion criteria include the presence of a known mild mutation, of which there are a number, clinical features suggesting a mild phenotype, such as a GMFM score, raw score of greater than 35% of goal, and any medical contraindications to surgery. So this table shows our patient characteristics, which, as you can see, are roughly half male and half female and span an age from 11 to 58 months at the time of treatment. The oldest and sickest patients were treated first. There was a hiatus in enrollment between patients 1, 2, and 3 and patients 4 through 8 due to vector production issues, and thus we currently have 6-month data on all patients right now, but are still collecting the 12 to 24-month data on later patients. As you can see, they come from all over the world, from New York to Alaska, and 5 foreign countries, including Chile, Argentina, Slovakia, Russia, and Italy. So I'd like to present some clinical vignettes from the Canavan Neurological Exam, or CNE, which includes videotape components. So this is a patient prior to gene therapy, that's me at the left, showing significant head lag and floppiness in the supine position. Then 3 months after gene therapy, the same subject shows improvement in head control, truncal stability, and gross motor function, including taking some steps with assistance. At 6 months post-gene therapy, at right here in this patient, we observed increased cognitive performance as well as the ability to stand from a sitting position and ability to stand upright without truncal support at the right, shown here with his physical therapist during a therapy session. So moving on to some quantitative data, grab the pointer. So starting here at the right, I'm sure this is working, but starting at the right, we have GMFM subscores of lying and rolling and sitting, which both show a favorable trend in the change from baseline raw scores. Then when the data are aggregated at left, the total change from baseline raw scores were statistically significant. This result is consistent with clinical observations of improved head and truncal control, which we believe are clinically meaningful. And we expect that these data will coalesce at the 12 and 24-month time points as we collect longer-term data on patients who were treated at a younger age. So the Mullen Scales of Early Learning is our other primary outcome measure, which tests a number of different clinical areas from the GMFM. It has non-overlapping gross motor function items as well as fine motor, visual reception, and receptive and expressive language. So these tests were done by a trained psychologist or medical doctor. And as you can see, we saw the same trend and improvement in gross motor scores as with GMFM, with a slight inflection point at 3 months, and then improvement in mean scores across all categories at 6 months. There was considerable inter-subject variability at baseline, but these data are represented as change from baseline scores for all patients. Of note, there were statistically significant changes in both receptive and expressive language at the 6-month time point, with some subjects showing emerging language and reasoning skills. As I mentioned before, brain imaging is a secondary outcome measure. We chose to use synthetic MRI or psi-MRI, a fully quantitative FDA-cleared method which uses a very short scan time to derive all the required sequences. It can precisely measure brain water content, myelin, white matter, gray matter, and other relevant parameters. Remarkably, Even with the limited data set at 6 months, we detected statistically significant changes in a number of key areas relevant to Canavan disease. There were large post-treatment water shifts measured both in CSF volumes as well as intraparenchymal water. The CSF volumes are shown at top left. In addition, we saw preservation of brain structure or lack of significant progression of brain atrophy evident with increasing brain parenchymal volumes and white matter volumes. Across the board on all patients, we observed increases in healthy myelin signal, which were on the order of about 10 mLs new myelin for all patients, which is, I believe, on the bottom left. So these are representative slices using synthetic MRI. They show brain white matter content for 3 different subjects at baseline and at 6 months post-treatment. The software sums over all slices, axial slices, to generate a total white matter volume for each patient. We observed variable increases in myelin across all the treated patients, which ranged from approximately 40% increases up to doubling of white matter content. These data, in my opinion, are extremely encouraging, using myelin as a direct biomarker of disease progression rather than an indirect biomarker such as NAA, which we're also collecting. Due to time considerations, I'm not presenting our quantitative NAA data today, but I'd just like to state that we've observed a clinically significant decrease of brain NAA with MRS on the order of several millimolar using single voxel MRS with our normalized LC model. In terms of adverse events, this is obviously an important part of a phase 1/2 study. There were no SAEs related to the drug, although 2 SAEs were probably related to the neurosurgical procedure. These involved a CSF leak and an episode of focal motor status epilepticus related to a catheter placement. We mitigated the risk of future SAEs through minor modifications of the neurosurgical procedure, including a change in the method of catheter insertion, changes to the skin closure, and measuring CSF pressures with pre-surgical lumbar drainage in selected patients. Some of the other SAEs included generalized seizures in some patients, which we believe are related to the natural history of Canavan disease, along with concomitant illnesses such as COVID or other viral infections. 2 out of 8 of our patients contracted COVID and recovered fully. So as PI and on behalf of Myrtelle, I'd like to thank all of our collaborators at Dayton Children's Hospital who made this study possible, including my co-PI neurosurgeon, Dr. Rob Lober, and his assistant, Tricia Ferris. I'd also like to especially thank Dr. Paola Leone and Dr. Jeremy Francis at Rowan University for this 25-year journey with Canavan disease, which we've been on together since we were at Yale together. Finally, I'd like to thank all the patients and their families for their resilience and commitment as we try to bring this novel gene therapy to the clinic. Thank you.
Thank you very much, Chris. Appreciate that talk. As the last presentation, we would like to invite Rick Lair up, and Rick is Senior Vice President of Research at Myrtelle, and he's going to speak on oligodendrocyte targeted gene therapies, new directions.
Thank you, Mark. So, for review, oligodendrocytes are the myelinating cells of the central nervous system. And myelination is the process by which oligodendrocytes extend membranes which wrap tightly around axons and form myelin sheaths. Myelin sheaths provide metabolic and trophic support to axons, but also, because they're separated by nodes of Ranvier, enable saltatory conduction, by which the action potential jumps from node to node, which greatly increases the velocity of action potentials, increases the distance that action potentials can travel, and reduces the energetic cost White matter, which is approximately half of the volume of a human brain, as Chris said, is composed of myelinated axons and enables the transfer of signals across distributed brain areas. Finally, as Ethan discussed, myelination is adaptive. Oligodendrocytes can adjust action potential velocity by changing the length of myelin sheaths, thickness, and by creating new action myelin sheaths. We've learned a lot about oligodendrocytes in the past decade because of the availability of new technologies and tools, such as single-cell profiling technologies, which enable the characterization of gene expression at the single-cell level. New MRI technologies, as Chris showed, enable the evaluation of white matter structure and volume. CRISPR-Cas9 gene editing systems enable the creation of new animal models of human genetic diseases. Two-photon microscopy, as Ethan showed, enables the imaging of fluorescently labeled cells in the living brain. Finally, the use of oligodendrocyte-specific promoters, or capsids, AV capsids like oligo-01, enable the targeted delivery of genetic cargoes to oligodendrocytes. This opens up the possibility of treating white matter diseases like the leukodystrophies, which are a group of neurodegenerative disorders that demonstrate abnormalities in myelin. Specific leukodystrophies are rare, but when combined, they show a prevalence of approximately 27,000 people in the United States, with significant morbidities and death in a third by the age of 8. They're characterized by progressive motor and cognitive dysfunction, with clinical symptoms appearing early in childhood, and abnormalities in white matter signal on neuroimaging. For example, this is an MRI from a 5-year-old boy with Pelizaeus-Merzbacher disease, and you can see here the hyperintensity on T2 that's not seen in the age-matched normal control. Recently, Josh Bonkowski and his colleagues at the University of Utah identified 399 genes that are associated with altered myelin on T2 MRI and are potentially causative for genetic white matter disease. For gene therapists like us, this is a lot of potential shots on goal. Specific leukodystrophies have the advantage as treatment targets because they're typically monogenic disorders. For example, Canavan disease that you've heard about is caused by a mutation in the gene for aspartoacylase. Hypomyelination with atrophy of the basal ganglia and cerebellum is caused by mutations in the gene for a microtubule protein. Krabbe disease is caused by mutations in the enzyme galactocerebrosidase. Pelizaeus-Merzbacher disease is caused by mutations in or duplications of the gene for proteolipid protein 1. Pelizaeus-Merzbacher-like disease is caused by mutations in a specific gap junction protein. Finally, autosomal dominant leukodystrophy, which is an adult onset leukodystrophy, results from duplication of the gene for a nuclear lamina protein, lamin B1, which disrupts myelin structure. Beyond the leukodystrophies, oligodendrocyte dysfunction appears to be playing a role in other neurodegenerative diseases like multiple sclerosis, which is the most common demyelinating disorder, with approximately 1 million patients in the US. The prevailing view of multiple sclerosis is that it begins with the peripheral nervous system in what some call the outside-in view. The immune system is provoked to initiate an autoimmune attack against components of myelin, which results in focused areas of demyelination called plaques, which can then cause neurological dysfunction and can potentially be remyelinated upon proliferation of oligodendrocyte precursor cells. But new studies are beginning to expand and even challenge this view. For example, Young et al. Looked at the integration of carbon-14 into genomic DNA from oligodendrocytes. Carbon-14 is a residue of the nuclear bomb testing in the 1960s. There was a lot of it then, there's not much now. So it's a good way to date cells. And what they saw was that most oligodendrocytes are long-lived post-mitotic cells. That's good, makes them a good target for AAV gene therapies. They also looked at oligodendrocytes in what are called shadow plaques. Shadow plaques are thought to be remyelinated areas that were former plaques. And what they found was that oligodendrocytes in shadow plaques are almost as old as the patient as well, suggesting that mature oligodendrocytes can contribute to remyelination in MS. Falcao and colleagues used single-cell RNA sequencing and showed that oligodendrocytes in brains from mice with experimental autoimmune encephalomyelopathy, which is a model of MS, expressed unique disease-specific genes that weren't seen in the control mice. Moreover, a substantial portion of MS susceptibility genes are expressed in oligodendrocytes. Jekyll and colleagues used single nuclei RNA sequencing and showed that, first of all, oligodendrocytes are heterogeneous in humans, like in mice, and that when they looked at oligodendrocytes close to lesions, they formed a distinct clustering profile that was also seen in oligodendrocytes that were distant from the lesions, indicating that MS is a more diffuse disease than the focal demyelination would suggest. Altogether, these studies indicate that oligodendrocytes are involved in MS and support an inside-out view and suggest that it's time to revisit oligodendrocytes in multiple sclerosis. Growing evidence implicates oligodendrocyte dysfunction in other neurodegenerative diseases like multiple system atrophy, which is characterized by the accumulation of alpha-synuclein in oligodendrocytes in the form of glial cytoplasmic inclusions. Jeffrey Kordauer's group was able to use the AAV capsid oligo-01 to deliver the transgene for alpha-synuclein to white matter oligodendrocytes in non-human primates, and they were able to see alpha-synuclein overexpression and demyelination similar to human disease. In Alzheimer's disease, neuroimaging studies implicate oligodendrocyte abnormality and demyelination in Alzheimer's disease risk and progression. And recently, Blanchard and colleagues used single-cell transcriptomics to evaluate brains with and without the E4 allele of apolipoprotein E, the gene for apolipoprotein E. And what they saw was that ApoE4 brains were associated with aberrant cholesterol deposition in oligodendrocytes and reduced myelination. Agarwal and colleagues used single nuclei transcriptomics and found an association between Parkinson's disease risk and oligodendrocyte-specific gene expression, suggesting a causal role for oligodendrocytes in disease etiology. Growing evidence implicates oligodendrocyte dysfunction in neurodevelopmental and psychiatric disorders. For example, autism spectrum disorder is often considered a disorder of neural connectivity, but neuroimaging studies show atypical myelination patterns, both reduced and increased white matter density in various brain regions from autistic patients. Recently, Fan et al. Showed a shared transcriptomic profile between mice with an animal model of Pitt-Hopkins syndrome, which is a syndromic form of autism, and postmortem brains from human subjects. In schizophrenia, neuroimaging and postmortem studies suggest abnormal oligodendrocyte function. Finally, in major depressive disorder, single nucleus RNA sequencing analysis reveals a prominent dysregulation in a subset of oligodendrocyte precursor cells, suggesting participation in the pathogenesis of depression. In closing, multiple lines of evidence implicate oligodendrocyte dysfunction in diseases like the pediatric leukodystrophies, Canavan disease, Pelizaeus-Merzbacher disease, Krabbe disease, hypomyelination with atrophy of the basal ganglia and cerebellum, Pelizaeus-Merzbacher-like disease, the adult onset leukodystrophy, autosomal dominant leukodystrophy, neurodegenerative disorders like multiple system atrophy, Parkinson's disease, and Alzheimer's disease, psychiatric diseases like schizophrenia and major depression, autoimmune diseases like multiple sclerosis, and neurodevelopmental disorders like autism. The ability to use AAV vectors to deliver genetic cargoes to oligodendrocytes opens new avenues for the development of novel therapeutics. Thank you.
So I'd like to thank the speakers for a series of great talks and really laying out the continuum from a deeper understanding of the basic biology of these fascinating cells to how you can target them, proving the concept that they can be effectively targeted in human clinical disease, and then, of course, what the blue sky view might look like in terms of involvement in other disease states, and really causes one to think about the typical relationship between these cells and other cells in the brain, and in fact, the entire kind of puppet master relationship that may exist as we see these cells exerting more and more effects and playing larger roles. At this point, I'd like to invite the speakers up, and we'll start to take questions. As you come up to the mic, if you could just indicate who your question is addressed to, please.
Hi, Rachel Salzman, Armitage Bio. It's maybe to the whole panel. Wonderful series of talks. So 2 questions. Number 1, can you comment, maybe the last speaker, about the potential utility of this in other leukodystrophies like adrenal leukodystrophy or metachromatic, where we're getting ex vivo data, but maybe if these were being delivered in vivo, you might get like quicker therapeutic effect. And then the second question is a little bit more to the earlier talks. Can you comment on either data you saw you might expect in Schwann cells?
Take that one.
Yeah, I think there are a lot of developments in other leukodystrophy programs. There's a number of other leukodystrophies that have specific gene therapies in progress.
Well, I was thinking, for ALD, where they're using ex vivo, but you've got to wait for those microglia to turn over, so your therapeutic effect is delayed by like 6 to 12 months. And I'm presuming this would be, delivered directly, so you might have sooner efficacy.
One would think.
Okay. And then about the Schwann cells?
I think, Rick, you can speak to the Schwann cells if you want.
We do see that oligo-1 is able to transduce Schwann cells when they're injected directly into nerve. So yes.
Thank you.
Please.
Hi, great talks. I'm Deepa Rao from Greenfield Pathology Services. My question is for Ethan Hughes. It's a more basic question on the methodology. When you use the two-photon microscopy method, what is the depth at which you're able to capture? Because the question relates to the kind of oligos, whether you're looking at the oligos in the gray matter versus the true corona radiata oligos.
Yeah, this is a great question. So for our two-photon imaging, we are currently imaging down to layers 4 and 5 of the cortex, which is about 400, 500 microns. But I think this is a really important point in that our studies that I talked about today are all focused in cortical axons and the oligodendrocytes that reside there. But there's a lot of studies, both pathologically in different disease models, as well as in the fundamental space that gray and white matter oligodendrocytes may have different transcriptional profiles as well as different microenvironments that they're acting in. And so from this motivation, we've now built a 3-photon microscope and done longitudinal 3-photon imaging to be able to look at the cellular dynamics of white matter oligodendrocytes compared to gray matter oligodendrocytes. And so I think these fundamental approaches may continue to give us more insights into dynamics of not just cortical oligodendrocytes, but also white matter oligodendrocytes too. Thanks.
Please.
I'm Hirokazu Hirai in Gunma University from Japan. Can you comment a little bit more about AAV Oligo 001? So you obtained that capsid mutant by capsid shuffling or something. So what type of capsid is that?
Rick, do you want to take that and maybe describe the derivation and the selection?
It was developed using capsid shuffling by Stephen Gray's group, who's here.
Oh, I see.
So it's a hybrid between a number of different starting points.
Okay. And another question is, in the case of clinical application, how many percentage of oligodendrocytes in the brain are transduced?
That's
Can you
yeah.
assume?
In preclinical models, we see a high percentage of transduced oligodendrocytes, but of course that depends on the route that it's given. But the papers published by Dr. Leone and Dr. Francis show over 90% specificity for oligodendrocytes.
Specificity is over 90%. How about the efficiency? How many? So let's say 100 oligodendrocytes. Then how many oligodendrocytes are transduced?
I don't recall the numbers of oligodendrocytes, but I think it was a high percentage.
Okay.
Well, it's process dependent, right? So I mean, it's how it's introduced and then flow rate, volume, all that titer.
Okay. Yeah. Thank you.
Yes. So Steve Gray, UT Southwestern Medical Center, and there's all the derivation of the OLG-001 capsid is published. It's pretty. You just search for it on PubMed. There's all the details are there, and the full sequence of it and everything. So, but I was going to ask to Chris Jansen. I mean, first of all, it's just exciting to me because. I mean, we made that capsid a number of years ago, and it wasn't directly what we were kind of focused on. It was actually almost a accidental byproduct of some other things that we were doing, and I'm so glad to see people using it and being productive with it. But the Canavan data, I mean, it's really the trial data is really striking, really amazing. Congratulations. Can you just educate me, I guess, about some of the natural history of Canavan disease? And the age that you're treating, like, would you ever expect a Canavan child to have those kind of increases in brain volume, increases in myelin volume, through normal development?
Yeah, no, we really haven't seen that before. The natural history is progressive brain atrophy. So anything that shifts that trend, or that slope in a different direction is very significant clinically. And as I mentioned, we're looking not just at the volume of brain, but also at the constituents like myelin and the water content, that sort of thing. So we think it's a real physiologic effect. Something good is definitely happening there.
And
But
it looked like you had an age range of between about 1 year old and 5 years old when you administered. I know it's only 8 patients, but generally speaking, did you see any kind of age-related effects?
Yeah, I think that ideally you'd treat them all quite young, under 12 months probably. But it's interesting, one thing we found that even in patients with pretty striking brain atrophy at the beginning, they also showed some of those changes, and that may have something to do with the porosity of the brain, which increases spread of the vector. We're not sure exactly why, but we saw similar trends in even more severely affected patients. So, but ideally, I think we'd treat them as early as possible. Yeah.
Hello, Yulia Grishuk, Massachusetts General Hospital and Harvard Medical School. It's a great talks. I have a question for Water. I hope I say your name correctly. Okay. So can you comment, please, on tools, which is a capsid and promoter combination for oligodendrocyte precursor targeting, and also neuron and oligodendrocyte duo?
Yeah, that's a really great question. And so we've been actually looking for any serotype or any cell-specific promoter that can give you any functional transduction in OPCs or any precursor cells. So it seems that it's actually very hard to target those cells. And even if you're using promoters that you would think that would target OPCs, you would still end up with transductional or functional transduction of mature oligodendrocytes. So for some reason, it's very difficult to target these cells. So if anyone would come up with a system, that would allow to target these cells, I would be very much interested in using these. As to your second question, so as to whether to target, so is that if you want to target neurons or oligos at the same time? I think there might be some systems. We've actually accidentally developed some promoters where you have sort of 60-40, 50-50 expression between oligos and neurons. So I'm happy to talk more about that and share some of that with you. And otherwise, I think you could always just try a mixture of broad tropism serotypes with different promoters, and just basically mix those and for instance, inject those or use those in your experiments.
Rick, Chris, did you guys have anything to say about the OPC targeting?
Well, we know from
Can you pull the mic close to you?
the paper from Dr. Francis and Dr. Leone that there was co-labeling with OLIG2 and BRDU, which labels proliferating cells. So it's likely that those were from OPCs.
Hi. Good afternoon. I'm Sherry Oban. I'm from UCB, but I'm also a rare mom who's a patient advocate. I wanted to build on the gentleman's question. So first of all, thank you for keeping the patient in mind and sharing some of those videos, because those are always so inspiring to see the changes in children. I have two questions. So the first question is, in the cohort, you mentioned you treated the most severe patients first, or the sickest. Can you give a little insight on kind of its heterogeneous and some of the symptoms in the group? And then my second question is on the procedure. What's that patient experience and what do families go through to get this delivered? Kind of.
Yeah, those are great questions. Maybe I'll address the second question first. So I think the patient experience, I think, has been very positive, and the patients and their families, mainly the families, are extraordinarily motivated, and they've traveled from all over the world and they've put up with a lot of testing. So I think The sponsors tried to make it as easy on them as possible in terms of travel and support and having a network at the hospital. So I think that's been gone quite well. Then in terms of the patient characteristics, yeah, there's some heterogeneity, I mean, but they all have the core features of typical Canavan disease. They all have poor head and neck control, big heads, the macrocephaly, the other features, spasticity, and other motor and cognitive features. It just so happened that the first patients we treated were a little bit older, but then there were older patients mixed in the bunch. So I think we got a good, range of patients, and I think everybody showed some clinical change that was encouraging. So.
Hello, Paola Leone here with Rowan Virtua. Just a question for Ethan, and the question is, you show remarkable changes, and also novel in terms of propagation and conduction and, physical changes in myelin during learning. And my question is, have you looked at motor function alone, separately from learning, to see if changes in myelination can be just affected by, because the testing requires movement, and so you're looking at learning as well as, and how would that just...
Yeah, this is a great question. And so one of our controls was mice that had learned the task 3 weeks prior to the onset of imaging. And then an additional 3 weeks later, we had them practice or rehearse the task. And we found that this did not change the formation or rate of new oligodendrocytes that are formed. We didn't do as careful of analyses of the change in the myelin pattern in that particular study, but I think that would be a very interesting thing to explore. But overall, our current thought in this space is that we think that it's the novelty of the learning itself that's involved in modulating at least the oligodendrogenesis responses. I think something might be also similarly occurring in the changes in myelination, but that's something we're actively exploring. Thanks for the question.
Question for Wouter. And the question is, so looking at the serotypes that specifically had the promoter selected for targeting oligodendrocyte, have you looked at the vector genome burden in other cells? Because the fact that you don't see the transgene doesn't mean that the transgene is not there, it's just not expressing. So in other
Yeah,
cells,
absolutely. So I mean, these vectors definitely get into different cell types, but we haven't exactly looked at the burden or at the transcripts that we see in these other cells. We certainly do not see any expression. So it's also why we talk about restricting the expression to oligodendrocytes as transducing these cells and seeing the transgenes there. But it seems that it's sufficient for these promoters to really drive expression specifically in these cells. It's kind of interesting when you start playing around with these promoters and try to shorten them and to get more packaging capacity. It also leads to more non-specific expression for some reason. So there's definitely some interesting regulatory elements there that are probably inhibiting and preventing the expression from of these transgenes in other cells. Yeah.
Excellent. I think that does it for the question. So I'd like to thank all of the speakers for today's presentations. It was excellent. Thank you very much. And also want to thank everybody who attended. Thank you for coming to this. Hopefully we achieved one of our goals, which was to teach everybody a little bit more about oligodendrocytes. So thank you very much.